My name is Dennis Hancock. I get to be the Center Director at the U.S. Dairy Forage Research Center here in Madison. We are part of the USDA Ag Research Service, and we have a group of scientists there, about 25 scientists that we have, as well as a number of staff and a research farm outside of Prairie du Sac. We also have a research group up in Marshfield in central Wisconsin at the research dairy up there as well. We're excited this morning about this first topic. I apologize, Gonzalo. It keeps going forward on the slides. This topic, I think, is one that is on everyone's mind these days about pre- and post-harvest practices to really maximize corn silage production and utilization. So Dr. Gonzalo Ferreira is a researcher at Virginia Tech, and we've asked him today to come and talk about some of his research, some really fantastic work, and I am excited to get to share it with you. So, Gonzalo?
All right, thank you. Good morning, everybody. So first of all, thank you, Dr. Hancock, for the introduction, and thank you for inviting me to present. Let me see if this is all right. So the idea today is to talk a little about the pre-harvesting and post-harvesting practices to maximize corn silage production and utilization. Hopefully I will make with the time. As you have seen already the outline, there is quite a heavy outline here. We are going to discuss a little about crop rotation and planting date, hybrid selection, planting density and nitrogen fertilization, the effect of drought on fiber digestibility, maturity at harvest, cutting height, kernel processing, and the ensiling. So, jumping right into the topic related to crop rotation and planting date. Again, the silage starts at the time that you are planting that crop, and I give a lot of importance to the rotation and the planting date, okay? Especially here, by the way, I'm originally from Argentina. In Argentina, there is a much nice weather than here in the US. So doing the double cropping, it's a little more gentle, but here, we have the windows that are narrower, so we have less time to do the double cropping. This is some data from the University of Minnesota, some years now, but it is good to describe as a concept the effect of the double cropping in the whole forage production. In this study, what they did is they planted corn at an early date of planting, okay? Then and there was no preceding crop. There was no crop before that. Then they did the same, but it was planted a few days later, okay? And here we have the yield, 18 tons of dry matter per hectare. And here it dropped a little. Statistically, was kind of the same yield, but there was a low trend to go down. However, then, when they did this other treatment, which is planting early, which is the same as this one, but in this case with rye as a preceding crop, they started to see a significant decrease in the yield of the corn, okay? And then when that, in addition to having a preceding crop and planted either later, the yield of biomass was substantially decreased, okay? This is in 1 year. The study was done for 3 consecutive years. And here we have the yield of dry matter. You can see this is the one I showed it to you before, going down with the later planting season and the use of the double cropping. The same, it was repeated here. Maybe the total tonnage here was the same. It was not different. However, if you see on the corn side, which is the orange bars, there was a depression on the yield of corn. And the same for the 3rd year. So again, managing the rotation, how are you going to grow the crop? And if you are going to do a double cropping, planting date should be key in your decision-making process. I'm going to discuss a little about the hybrid selection. There is more and more variety of hybrids that we can select for silage. And I think that is going to keep changing, adding more and more materials. To me, this is a key concept that I like to students to understand, and hopefully farmers as well. And that is what we call the Lucas test of forage quality. What it says, the Lucas test, is that mainly we separate the forages in two components, cell contents and cell wall, okay, or fiber. And the important thing here about the cell contents, here we have this line. Here we have what we call the neutral detergent solubles. Those would be the cell contents of a forage. And then here we have how much of those were digested. If you see the slope of this line, which is this number here, is 1. What is the meaning of that? Is that the digestibility of the solubles is pretty much 100%, 98%. And then if you see all these dots here, pretty much they're around that line. That means that pretty much always they're going to be digestible in 100%. So bottom line, cell contents, they digest completely and very uniformly, okay? Which ones are some components of the cell contents? Typically, of the solubles, starch, okay? So starch typically is going to be very highly and uniformly digested. In the case of the fiber, that is going to be totally different. Well, not totally different, but it was not going to be as nice as here. First of all, the slope is not going to be 1. That means that it's not going to be completely digested. And also there is going to be much more variation in the digestion of that fiber, okay? If you go there and you see the NDF digestibilities that you see there on the corn silage, typically those values are around 70%, okay? So bottom line, more solubles, more digest, and the energy that is going to have that silage. And I think we all know that part. Oh, geez. In that hybrid selection, obviously, we have many options. Typically, we say conventional when there is no specific traits. On the endosperm type, we can talk about dentate versus flint, which is related to how hard is the starch in that kernel. Also, the waxy could be on the grain side. Brown midrib, which means the hybrids that they have less lignin concentration in the whole plant and also in the fiber, and therefore ends up with greater fiber digestibility. The leafy materials, those are plants that they have more leaves relative to the stems, and therefore they should have a difference in the digestibility of the fiber, high oil, high lysine, and so on. About brown midrib, I think most of the people know what brown midrib is, but just in case, brown midrib is a mutation that inhibits a pathway in the synthesis of lignin. Lignin is a component of the fiber that avoids the degradation of the fiber, okay? So a corn silage that has a greater concentration of lignin is going to be less digestible than fiber. Well, there is data showing that the varieties with the brown midrib, they have an increase in milk yield, as you can see here, and also an increase in dry matter intake. Because the cow eats more, the production ends up being higher. And this is quite consistent about different studies. What is the reason for that increase in milk yield? Well, very consistently, this is a study we have done a few years ago in Virginia. This is an on-farm study done in a dairy farm, commercial dairy farm. And here we planted 2 fields, field number 1, field number 2, at 2 different years, 2017 and 2018. and we planted the BMR and a conventional corn at 3 different planting populations. Consistently, if you see the yellow bar, the lines here, which is the BMR, consistently in all this figure is in vitro fiber digestibility. The BMR has been 10 to 15 units greater in NDF digestibility. This is very, very consistent, okay? Now, in this same study, one drawback that we have seen is that here we have dry mature yield in the same fields, same years, and so on. We have a flip. You should expect a reduction on dry mature yield by using these materials. Another thing that is, at least to me, it was kind of surprising, is not thinking on the leaf-to-stem ratio in corn as we do it with alfalfa or some other grasses. Typically, if we think about alfalfa, the goodies are in the leaves, right? And the poor quality is in the stems. And therefore, we say an alfalfa with a greater leaf-to-stem ratio is going to have a greater dry mature digestibility. And that is correct. The same happens with the ryegrass. The ryegrass is an excellent grass until it grows and it has the stem, right? So what was surprising to us, we did a study in which we measured the NDF concentration, the fiber concentration in the leaves and in the stems. And somehow we saw that there was we saw that there was a greater concentration of fiber in the leaves than in the internodes, okay? And that goes against what we learned with alfalfa and ryegrasses, okay? Or other type of grasses. So that was kind of shocking. This was a study that we did all this. We also did a drought study. We observed the same thing, greater concentration of fiber in the leaves than in the internodes of the corn. So, how can we explain that greater concentration of fiber in the leaves than in the stems? And this is what we proposed. We just published a paper on this, and I think it's related to the structure of that stem. If we think about ryegrass, barley, wheat, oats, all those winter grasses that you are familiar with, they have a hollow stem, okay? And typically, that stem has a very high fiber concentration. In the case of the corn or the sorghum or the pearl millet, we have more a non-hollow stem. Now, look at this inside of the internode. I like to call it, well, I had an image. If you see on the microscope, inside this stem, there are a lot of cells with very thin cell walls. And those cell walls, I like to say, when I ask the technician, they look like a potato, if you want, okay? So I said to the technician, okay, let's do the fiber on the outside of the stem and the inside. And this is what we got. I'm sorry. Okay? We took the core of that internode and we did the fiber on the cortex and on the pith, on the inside. Okay? And this is much more similar to a typical stem of a ryegrass or a barley or something of a hollow type of grass. Okay? And we have this with only 35% NDF. So far, this is kind of our best explanation for looking that inversion in the concentration of fiber between the leaves and the internodes. Now I'm going to talk a little about planting population. I thought that we were doing research with a lot of plants until I heard that you're using 160,000. Anyhow, this line of study was motivated by Alberto. He was a dairy farmer from Argentina, and I was working with him, and at that time he was putting a lot of plants in the field, and I was saying to him, you're going to go off the road, you know, you're putting too many plants. And he liked that idea. Anyhow, we did a study, and we planted here you have the different populations, 60,000 plants per hectare, 70, 80, or 90. And here you have the weight of the plant, okay? And we use two different varieties of corn. This figure, what it's saying mainly is that the weight of the plant was not affected by the planting population, okay? Now, when you plant at different planting populations and you have the same size of the plant, what is going to happen? You're going to have more plants of the same size, so you're having greater yield. So here you have how the biomass was increased by increasing corn planting population. What was curious on that study is that the fiber concentration of those plots was not affected, was the same NDF concentration. So somehow similar fiber, similar quality. The starch also, if you see within the varieties, it was not different among the different planting populations. So somehow the message here it was, okay, you put more plants, you can get more biomass, and you are not really affecting the quality of that forage. Then I came to Virginia a few years ago, 9 years ago, and I started to ask the farmers, okay, how many plants are you putting in your field? And some people said 65,000 plants, 70,000 plants, 80,000 plants. And I said, okay, what is the target here in Virginia? How does it correlate to the study that we did in the past? One thing about the study in the past is that it was corn planted on top of corn for grain. So there was a long resting period, remember about the rotation, between one crop and the other, okay? So there was good accumulation of water in the soil, and it was good accumulation of fertility in the soil. It was not a double cropping situation. So here we did a study in Virginia with a double cropping situation. And in this case, when we put more plants, as you can see here, the weight of the plant went down, okay? So it was more challenging with the higher planting population. However, still, we saw that increase in the biomass production, okay? So by increasing the corn planting population, you can get more silage from your field. Let's see what happened with the quality. Again, fiber concentration here as a percent of the dry matter. There was a linear effect, but if you think about this, less than 2 units of fiber difference. So a tiny difference here in the concentration of the fiber. And then the fiber digestibility, statistically, there was no change by increasing the planting population. So far, yes, we are having smaller plants, more plants, so therefore more yield, a slight increase in the fiber concentration. Now, when you saw the crop, this is what we have seen, and this is like for a book. It was perfect. You see a very nice, at low planting population, a very nice and big ear full of grain. And when you have the increased planting population, you start to see that tip of the ear with less kernels, okay? So if you see this, you would say, okay, starch concentration should go down. And the thing is that we did not see that. I thought I had the starch concentration there. But anyhow, what happened? What we did in that study, we counted the number of lines of kernels that we have in that ear, okay? And there was a significant decrease in the number of lines that you have in that crop. To give you an idea, these pictures are from here when the ear is this size, okay? And that is when the kernels are being differentiating, okay? So typically, when you increase the corn planting population, you might be affecting the number of lines that you have in that ear. And in addition to that, you might be affecting the potential number of kernels in that line, okay? So far, we had less number of lines per year. Also, we counted the number of kernels in those lines, and the number of kernels per line went down, okay? So that is why we had less kernels in those years, when you go at the higher planting population. What is the result? You have less lines, you have less kernels per line, you have less kernels per year or per plant, okay? Now, what happened? The starch was not that different. We had less grain, but we had a similar starch concentration. So we also measured the width of the stem with a caliper in this case, and we also found that the width of the stem is reduced by the high planting population. Bottom line, you do have a lighter plant with smaller ears, but also with smaller stems, okay? So somehow they're kind of proportional. But we are still having more yield, more biomass. Somebody told me, well, when they saw those pictures, oh, that is a problem of nitrogen. And it could be, it makes sense. This is a typical sign of nitrogen deficiency. So then we did another study, also in a dairy farm in Virginia. And what we did is we doubled the amount of nitrogen at V6. V6 is when the plant has 6 visible leaves, and that is kind of where that ear starts to differentiate. So at the end, he worked on his nitrogen plan, and at V6, we did half of the plots, 1 pass of nitrogen, and the other half double pass of the nitrogen with liquid nitrogen. Dry matter yield, we saw an increase in dry matter yield. This was 0.6 tons per acre. The plant weight was slightly greater, but not statistically significant different. However, with more nitrogen, we could increase the number of kernels per year. We also increased the stem width. However, we did not see any differences in the composition of the biomass and on the digestibility of that fiber. All right, now I'm switching gears to drought stress. That is a main interest that I have, especially after working a lot in my years in Argentina, 5 years, 3 years of drought. So there is a concept that heat stress and drought typically increase fiber digestibility. In this case, they said something like the good part of it, but reduce yield. I agree with the yield part. I'm not very sure with the increase of fiber digestibility when there is a drought, okay? To me, it doesn't make a lot of sense. So we did a study a few years ago in 2018 with my colleagues from University of Idaho, and we planted 5 different varieties of corn, and for that we use an irrigation system. In Idaho, they irrigate by flooding, so they release the water, right? And we, first of all, doing a drought stress study, control study is super difficult. But anyhow, this is what we got. And we were quite successful. You can see here the senescence of those leaves that they were not fully irrigated. What have we seen on the quality of those forages? First of all, in this case, the concentration of fiber, here I'm not talking about digestibility, I'm talking concentration of fiber went up. And somehow to me makes sense. If you remember those senescent browning leaves, that means that there is more there is more fiber and less cell content, less juice if you want, okay? So fiber should go up with drought. What happens with, in this case, we have the in vitro NDF digestibility. Here there was a tendency for fiber digestibility to go down with the drought, okay? So that goes against that message that we tend to hear, okay? And then in the case of the lignin, tended to go up. There is more slightly, very little difference here, okay? But more lignin in that drought stress corn. So bottom line, we need to work a little more on the effect of drought on fiber digestibility. I do agree, especially those of you who harvest corn. When we have a drought, a severe drought, one thing that we tend to do is to panic and we go and harvest early, okay? That might be the reason why we are getting a higher fiber digestibility. This is a typical question I ask in class and many times among farmers. If we have this field of corn and we harvest 50% today, and then we have a rain or whatever, the chopper breaks, and we need to go back in a few weeks, okay? Which one is going to be the one that is going to have the greater fiber concentration? I will not do the test here, but typically people say that this one harvested later is going to be is going to have a greater concentration of fiber. That is not the case. Typically, when we harvest later, we have more grain in that whole crop, and that grain dilutes the fiber of the vegetative portion, okay? So do not think that because you're harvesting later, you're going to have more fiber in that silage. Likely everybody knows about harvesting time, when to approach the field for topping. I prepared this for class, mainly for the students, but typically I tell them go to Kroger, to the supermarket, buy a corn. If you press the kernel and splashes your face, that is not ready, okay? So that is the number one. When you start seeing the dent, you have an early dent there, you can start thinking about harvesting, and then you can look to the milk line, which is the separation between the dry endosperm and the milky endosperm, and you go from there. And then also you need to identify what is the black layer, which is you peel the tip of the kernel, and if you see this black tissue there, that means that that is the black layer. Here you have a comparison of the black layer stage and a milky stage. Bottom line, look at this. Here we have the chemical composition of different corn silages that they were harvested at early dent, 1/4 of the milk line, 2/3 of the milk line, and the black layer stage. And as I told you, if you think about it, this is early maturity, this is late maturity. Look at the NDF. It went down over time, okay? And if you see the starch, it went up over time. So again, late maturity, the whole crop is going to have less fiber, okay? It's going to have more energy because there is more starch in the proportion of that. The question is if that starch is going to be digestible or not. Here we have the chemical composition of the stover. The stover would be the stem plus the leaves, okay? And in this case, when you see the stover without the grain component, then yes, with advanced maturity, you have a greater NDF concentration. The thing is, when you have that grain component, you are going to have a decrease in fiber concentration. And of course, with more time, you are going to have a decrease in dry matter digestibility of that stover. what is the effect on the diets? Here, that same corn, I showed it before, harvesting at these different times. They measured the digestibility of the fiber and the digestibility of the starch. And you can see here, fiber digestibility always was going down, okay, over time. So that is the bad side of waiting late maturity corn. However, in the case of the starch, here it was 94% digestible, here 93. Up to 2/3 of the milk line, you start to see the bad consequences of waiting too long. So you should not go to more than 2/3 of the milk line. Bottom line, starch digestibility decreases with maturity, especially after 40% dry mature. You have a greater proportion of the hard endosperm, harder kernels. They're more difficult to break during the harvesting and the chopping. And one of the reasons for that also is that there is a protein matrix that protects the starch in the grain, okay? That protein matrix is stronger with the late maturity. The recommendations, typically 1/2 to 2/3 of the milk line. This would be similar to a 35% dry mature concentration. I like to say 33 to start, so when you finish, you are closer to an average of 35. Try to avoid the 40% dry mature concentration so you don't have any packing problems, poor quality. And also try to avoid less than 30% dry mature concentration because you're going to have lower yields, seepage losses. The digestibility is going to be very good though. This is very fast, but in a case of a very late, you can see a lot of whole kernels in this silage, and it was super hard to package. That was harvested too late. This was harvested extremely early, okay? And this was to the point that the biomass was rolling on top of the bagger, okay? A little about chop length. We want to have physically effective fiber, so we stimulate the chewing activity in the cow. So we want long particles, also to induce that rumen mat in the rumen. However, we do not want anything excessive long. It's going to slower the passage rate, and you are going to start having sorting problems. As we can see here, a lot of whole cobs, okay? The ration maybe was formulated one way, but the cows were not eating exactly what it was formulated. So with the chop length, it's hard to give a specific recommendation. Usually you have to see what you're chopping. I like to say start with 3/4 to 1 inch of theoretical length of cut, search for round cup pieces, whole kernels, and then adjust as you can. And obviously, finally, you don't want to see this. This is not only from the silage, it was also the grain of the diet in this case, but put kernel processors to break that grain and increase the digestibility of the starch from that silage. If you put the kernel processors, obviously, you have to put them, but close the processors in the way that they do the work as they should. Here there is some data, for example, no kernel processor with 80% starch digestibility. Here they put the processors at 2 millimeters and they increase the starch digestibility, but when the processors were there but they were open, the digestibility was not as high as before. So again, put the processors, but put them and close them. And then to finish here, the cutting height. If you want to go for quality, obviously you can raise the bar and cut more. There are certain situations in which you need to get the most from your field, so there is a lot of argument there. The thing is, yes, you can increase the cutting height, and by doing that, you are increasing the proportion of the ear. You should have a forage with greater energy, but you're going to sacrifice the yield. We did a study in which we measured the NDF digestibility of the very bottom internode, and 3 internodes above, and the one in the middle. The concentration of fiber doesn't change that much over the plant. The digestibility of that fiber does differ, okay? But not that much. So bottom line is, this internode here, yes, it has poor quality, but this internode there is not much greater on a fiber perspective. All right, I'm already 5 minutes late. So in summary, look to your crop rotation with a particular importance to the planting date, hybrid selection. There you have to address what are the goals of your farm. Planting density, we can get more silage by increasing planting population. If you do, work on the fertilization. Drought stress and fiber digestibility, I think we need to work a little more on that. It is not clear cut. And then maturity at harvest, aim to 35% dry matter on average, and you should be having a good quality forage. Cutting height and kernel processing, obviously, things that you can work on that. That was a lot of information, I think. I thank you for your attention, and I would entertain any questions.
Hey, any questions for Gonzalo?
Yes.
How much yield do we give up between 6 inches and 20 inches?
Sorry, this is keep rolling. Yes, so the question is how much yield we sacrifice between going with the low cut and the high cut. As far as I remember from the literature, you were talking about 6% to 15% is the yield that you are sacrificing by going higher. Within that range, no more than 15. Yes, sir. I cannot hear you, sorry. Chop size. I was telling you, typically, that is something hard to recommend. Yeah, I think you have to be in the field to really adjust, in my opinion. I said 3/4 to 1 inch as a starting point and playing around with the kernel processor. Okay, if you're chopping too fine, maybe you have to go more towards the 1 inch. If you are getting too big of cobs, typically, maybe you have to start bringing it down. I don't remember, to be honest, the data on the digestibility of the fiber. I don't think it should affect that much. Obviously, if you are chopping too much or you're having highly fermentable starch, you might have a slight decrease, actually, on the fiber digestibility. I think there is some data from Dieman in Wisconsin that they actually show a slight decrease on the fiber digestibility. But I need to confirm that. Any other questions?
Gonzalo, just one comment about the drought stress corn. Sometimes that's going to be cut at a little higher height to avoid nitrates as well, right? That could be a factor in that digestibility.
Yes, I never thought about that, but yes, you're right. If you are concerned with the nitrate because they are at the base of the plant, you might increase cutting height, and therefore you can increase the fiber digestibility. Good point. Yes, very good. Thank you.
We have additional presentations throughout the week. I also would encourage you to go by our booth back here and pick up these little business cards that on the back of them have the links to the video recordings that will be made. So all these videos, all these presentations will be recorded and placed on the YouTube channel. So I encourage you to check that out as well. Thanks again, and I'll see you hopefully at 1:30.